chapter 12. I'm sure most of you are going to be excited to be finishing up this chapter. All right. So the last part that we're going to be talking about, we started talking about the different classifications of the neurotransmitters. And now we're going to finish up with just a couple more things on neurotransmitters, and then we're going to talk about this concept of neuromodulation. Okay? So keep in mind, when we're talking about the classifications of neurotransmitters, we classify them by their function. Okay? How they're going to affect that membrane potential. Kind of like the EPSPs and the IPSPs. Remember, the EPSPs decrease the membrane potential, right? Because they would allow for sodium, all right, a cation to enter into the cell, making the inside of the cell more positive or less negative. So we go from that negative 70 resting membrane potential to negative 68, negative 65. Okay? So that's pretty much what the EPSPs did. The IPSPs would increase the membrane potential charge in negativity. So it would take the resting membrane potential from negative 70 to negative 72, negative 75. All right. So that's how we classify, all right, our neurotransmitters, right, as excitatory or inhibitory. So that's a binary classification. Okay. It's either, you know how, well, I'm not big into coding, but binary coding is zeros and ones. That's all it is, all right? Or yes and no. That's another way of a binary classification. So with neurotransmitters, it's excitatory or inhibitory, okay? So keep that in mind, all right, when we're discussing that some of those transmitters, all right, are going to either excite some of our target cells or they're going to inhibit some of the target cells, and it really depends on what neurotransmitter is being released, right? So it's going to be specific or receptor specific, right? Depending on what neurotransmitter is going to be released, okay? So when we talk about this classification, when we talk about the classification of the action, we'll either have a direct or an indirect transmitter that's going to occur. Now, you'll get into this in a little bit more detail, all right, in 211. I'm just going to touch on the real basic. It's real simple for direct versus indirect. Then you get to learn all the different steps for the indirect. But for the direct, we've already talked about the direct, okay? Neurotransmitter gets released. It's a chemical. It binds to a chemically gated channel, right? And then that chemically gated channel opens up, and an ion or some substance of some sort enters into the cell or leaves the cell, okay? Something's going to cross that channel, right? So you're going to have an immediate response there. Okay. With the indirect, now we get into these secondary messengers and what we call G proteins. Again, it would take me several minutes to discuss that. I'm not going to do that because, like I said, we're going to learn it all in 211. All right. But what I want you to understand is with the indirect, all right, you're going to deal with the substance called G proteins and secondary messengers or second messengers. Basically, what a secondary messenger is it's like having a butler. Okay, I come to your front door, I knock on the door, all right, you're up putting your makeup on or getting dressed or you're in the shower or whatever, and I come to the front door and the butler answers the door and I said, listen, I've got to talk to so-and-so. Well, they're not available, but give me the message. I'll take the message to so-and-so and then tell them. That's essentially kind of like what a secondary messenger is, okay? So with that, we see a much more diverse type of effect occurring when we're dealing with the secondary messengers. A lot of hormone action occurs that way. Insulin for one, right? But again, that is for another time. Right? Figure 1224, this will show you the classifications for our neurotransmitter. One is based on the chemical structure, which we talked about last class, all right? Big acetylcholine, that's going to be seen for all our skeletal muscles. And when we get into the autonomic nervous system, we'll talk about that too. All right? And then about the function, and that's what we just talked about, the effects and then the actions of those functions. It's either going to be excitatory or inhibitory, right? Which will cause our, if it's excitatory, we're generating EPSPs. If it's inhibitory, it's going to be generating IPSPs. Then our actions are going to be direct, opening up a channel, and then ions can cross over the channel, or indirectly use G proteins or secondary messengers, okay? All right, so the four primary classes of neurotransmitters, you want to be familiar with these, all right? Especially this one here, because we're going to talk a lot about acetylcholine when we get into chapter 10, right? But we got acetylcholine, amino acids, right? Monoamines, and our neuropeptides. Those are your 4 primary classes of the neurotransmitters, and then there's subclasses to those, okay? But those are the 4 that you want to know. Right? Like I said, we talked about those last class. All right, so big neurotransmitter right here, acetylcholine, right? So a couple things you want to know about it. One, okay, it's used in the peripheral nervous system. PNS stands for peripheral nervous system, right? And that's going to stimulate our skeletal muscle, right? So, which is perfect, okay? Because that's going to be a primary or the neurotransmitter that we're going to discuss, right, quite a bit in chapter 10. Right. Also, we'll see in the central nervous system, and it talks about increasing arousal or awareness. All right, and we'll talk a little bit more about that when we get into the autonomic nervous system. All right, when we talk about sympathetic nervous system versus the parasympathetic nervous system. All right, so more on that later. But here's the thing: the 2 ingredients that we need to make acetylcholine is going to be acetate and choline. We'll combine those together to make acetylcholine, because what happens, and this we'll talk about in a little bit, there's an enzyme. All right, it's listed here on the slide, acetylcholinesterase, which breaks down acetylcholine. So basically, there's different ways, and we'll talk about it in a moment. When acetylcholine, the neurotransmitter, gets released from the synaptic knob, and it's floating around in the synaptic cleft. Eventually, it's going to stop working. Right? It's going to stop affecting, right, either the postsynaptic neuron or the effector organ, the target cell. Right? It's going to. One of the ways that we can cause it to stop doing that is if we break it down. If we break down acetylcholine into acetate and into choline, then the effects of acetylcholine aren't happening anymore because we no longer have acetylcholine, okay? So the enzyme that breaks it down is, you got to know this, right, acetylcholinesterase, because there are certain toxins that can actually cause acetylcholinesterase. You can block it, right? You can, if you have a drug that is able to inhibit acetylcholinesterase, then acetylcholine stays in the synaptic cleft longer, and you can prolong the effects of that neurotransmitter. So there's going to be certain drugs that will help with that if you want to sustain a muscle contraction, for example, right? So when we have acetylcholine, all right, released, all right, we need an action potential. So 2 ways, right, to prevent that neurotransmitter for acetylcholine, right, from being operable is 1, stop the action potential, don't stimulate the neuron, right, or 2, right, you can increase the acetylcholinesterase and it will break down the acetylcholine, okay? So that's what we're going to see in the synaptic cleft, right? Acetylcholinesterase comes in and it breaks down acetylcholine into its original components, right? Acetate and choline, okay? Then what will happen is that presynaptic neuron will reuptake or resorb, right, that acetate and the choline, and it'll make acetylcholine again inside the neuron and store it in the synaptic vesicles. And it'll wait to release that acetylcholine, right, when another action potential comes down. Okay? So keep in mind, and again, I don't get into this too much, right? But acetylcholine is going to affect the effector cell, right, differently depending on which type of receptor that we're going to be using. If it's a nicotinic, you guys are familiar with nicotine, okay? That's why it's one of the very addictive substances. You have specific receptors that are created, that are present in your body that are specific for nicotine. That's why it is considered to be one of the most addictive substances on the planet. And then you've got muscarinic receptors. Point being is, right, these different receptors will have, depending on which receptors are affected by the acetylcholine, will vary the effect on our target cell. Okay? And that is dependent upon which receptor you're activating. Okay? Right. That's acetylcholine. Right. So one of the ways that we can get rid of a neurotransmitter in the synaptic cleft is what I said before. Right? We have an enzyme there that's going to break down that neurotransmitter. Okay? That will stop the effects of the neurotransmitter. Another way to stop the effects of the neurotransmitter is through what we call reuptake. Right? That presynaptic neuron, right, is going to kind of vacuum up that neurotransmitter that's in the synaptic cleft. It'll resorb it and put it back into the presynaptic neuron, back into the synaptic vesicles there, all right? Or you will have, this one is not as popular, right? But that neurotransmitter will just diffuse away from the synapse, and our glial cells, all right, will absorb it up, okay? And that's 3 different ways, all right, that we are able to remove the neurotransmitter from the synaptic cleft and to stop its activity, okay? So we'll see some drugs that have a very similar type of response here, right, or effect of the things that I just mentioned here. SSRIs, selective serotonin reuptake inhibitors, right? This is the 2nd generation type of drug, right? So Zoloft, perfect example, right? Very popular antidepressant. Usually people that have depression, right, have low serotonin levels. Serotonin is a neurotransmitter, okay? So one of the ways that we can help promote serotonin's effects with the neurotransmitter is to prevent it being removed from the synaptic cleft. So these SSRIs, these selective serotonin reuptake inhibitors, prevent this step from happening, right? It inhibits these transporters here, right, in the presynaptic neuron from reuptaking the serotonin. So it stays in the synaptic cleft longer. So it can exert its effects longer, okay? This is a relatively new drug, all right? But this hydrobromide drug, all right, is an acetylcholine esterase that's a long-term inhibitor. So now we're preventing the enzyme, right, acetylcholine esterase, like I was telling you before, right, from breaking down acetylcholine. So acetylcholine stays in the synaptic cleft longer, right, and it can exert its effect. So we're finding that very useful in Alzheimer's disease because it affects the neurons in the central nervous system. Okay? There's all sorts of fun experimental drugs that we're using. All right. So this is basically showing you, all right, how acetylcholine here is getting released, right, from the presynaptic neuron. It diffuses across the synaptic cleft, and then it binds onto these receptors here on these channels, right? And then it causes the channels to open up, and it causes the ions to diffuse across the plasma membrane, okay? So here's well, it's tough to see there. This is an example of direct, okay? So here's the direct effect, right? The channel opens up, sodium enters in, right? Causes an EPSP. The indirect effect is it causes stimulation of our G protein here. That's our secondary messenger. And then there's a whole process with a G protein. I always say it skips on down the inside of the plasma membrane. Then it'll activate some other enzymes to do their thing. It's a whole series of steps, right? But that's the indirect. This little box here is explaining to us, all right, you have acetylcholine here, right? Then our acetylcholine esterase comes in, and it breaks it apart into choline and acetate. And then that acetate and choline get reabsorbed through these transporters back into our presynaptic knob. Okay? All right. Finally, the last part of chapter 12, neuromodulation. Okay? So basically what a neuromodulator does is it's going to somehow affect the response of neurons, okay? It can either increase the effect or the response of a neuron, or it can decrease the response of a neuron, right? When we talk about the first one, facilitation, we have a greater response, okay? When we're talking about inhibition, I don't think I need to explain to you, but it causes a weaker response, okay? So in this case, when we're talking about facilitation, we'll increase the amount of neurotransmitter that is released from the presynaptic neuron that goes into the cleft, right? And then therefore, if there's more neurotransmitter, right? We talked about this. We were talking about graded potentials. One of the ways that we can increase the graded potential is if we release more neurotransmitter and open more gates, which will let in more sodium for EPSPs. right? And that's what we're seeing here for this facilitation. We're going to increase the amount of neurotransmitter that we release from the presynaptic neuron. It diffuses across the cleft, and it's going to increase the number of postsynaptic receptors that will be affected. So we refer to that term as facilitation. With inhibition, just the opposite. We decrease the amount of neurotransmitter that's released from the presynaptic neuron, right? Therefore, there's less neurotransmitter in the synaptic cleft. Therefore, we decrease the number of the postsynaptic receptors that are affected. Very simple. Okay, it's just trying to memorize the term. Okay, so facilitation and inhibition. And that works out well for our graded potentials, because remember, the differences between graded potentials and action potentials you definitely need to know. All right, good. That didn't skip away. This slide here, and we talked about it. Where is it? This one right here. Boom. You guys already have notes on this, right? But you need to know the difference between graded potentials and action potentials. Okay? Remember, our action potentials, they're all or none. We can't increase the intensity of an action potential. Okay? Graded potentials, we call them graded potentials because they are what we refer to as being graded. So what does that mean? Right? The larger the potential change, that means if we go from a negative 70 resting millivolts to a negative 60, that's a pretty large response. All right? That's a pretty large change in the potential. If we go from a negative 70 to a negative 68, that's not very big. All right? So the larger the potential change, the stronger our stimulus will be. Right? That's like throwing a bigger rock into the pond. Okay? So if we throw a little pebble in, it's not going to be very big. If I throw a big 20-pound rock into the lake, it's going to make huge ripples. That's going to generate a larger response. All right? A stronger stimulus. Okay? Action potentials are all or none. So once they start, it's not going to be big, it's not going to be small, it's just going to be, all right? There's no big or small version of an action potential, okay? So you definitely want to know those differences. It'll make a huge, I don't want to say difference, but, all right. So last slide here, when we're talking about neuromodulations, you folks familiar with nitric oxide, okay? I'm sure you're probably familiar with the gas portion of it when you go over there to have to the dentist, okay? If you've ever gone to a Grateful Dead concert, there's people walking around with balloons sucking on this stuff, all right? Point being is, all right, in the peripheral nervous system, all right, nitric oxide, they used to use this stuff quite a bit in weightlifting, bodybuilding. You would go into like GNC or Vitamin Shop or Vitamin World, any of those places that would sell supplementation, and they used to advertise the heck out of this stuff because it causes the peripheral nervous system, right, blood vessel dilation, increases your blood flow. Okay, so when you're working out, lifting weights or running or whatever, you're going to increase the blood flow even more to the muscles, so they have more energy. Okay, so keep that in mind. This one here, the endocannabinoids. We're getting a lot of word on this stuff. You guys heard of CBD oil, right? Okay, that's a cannabinoid, right? So this stuff here, you've got these specialized receptors in your body, and in fact, these acetylcholine, which are locally acting hormones that your body produces, right? It helps with this neurotransmitter release here. Okay, there's several different effects here, right? But what we're talking about, these endocannabinoids, all right, are going to affect, all right, the presynaptic neurotransmitter release. In most cases, they can increase the presynaptic neurotransmitter release. But going back to what marijuana does, all right, it increases appetite. It's one of the effects. That's why it's a great drug to use on people that are suffering from the bad effects of chemotherapy, right? Because it helps increase their appetite when they're what we call cachectic, which is a death-like appearance. Some of the adverse effects of chemotherapy and even just of the cancer itself, it can kill somebody's appetite, and they found that medical grade marijuana helps to increase that. This is very PC here. It influences memory, all right? lot of pot smokers, and I'm not judging pot smokers, all right? But and a lot of my friends in high school did pot, and so they didn't have the best memory. They insisted that they studied for a test high, that they had to go and take a test high. I told them that was the worst possible rationale that you could use, because it's going to influence your performance, all right? But no one listened to me. All right, so the term of neuromodulation that results in a greater response in the postsynaptic neuron is facilitation. The lesser response is inhibition. Okay? When you're inhibited, you're less likely to do something. That's how I remember that. Right? Although alcohol does take away your inhibitions, so there's that. So you might be more prone to do something, all right, involving alcohol. All right. So that's the end of chapter 12. I know that's a lot. In chapter 12.